Hi everyone, are we live? Am I audible, visible? Am I audible, visible? Hi everyone, happy morning. Welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma. And today it is the part 2 of sexual reproduction in flowering plants. And tomorrow there will be the part 3. So in 3 sessions, we are going to finish this particular chapter. Okay? Done. So I hope you people will revise NCERT and will practice MCQs as well. And do let me know which chapter you want to start next. Okay, so let's start the class, students. And first of all, just have a look. Yesterday I posted this for all of you, right? I think there is some confusion related to the new NCERT QR code. Okay, so that is what I have discussed here, and after watching this video, everything will be crystal clear to you that which NCERT you need to follow, right? And is there any chances of syllabus deduction and not? Okay, so that will be very good. Do check it. If you have checked it, good. Otherwise, do check this particular video. And moreover, students, please do not waste your time okay, so do not waste your time, and please join HSP sir's mind map batch as soon as possible, because soon we are going to close the enrollment for that particular batch. And if you're facing any problem to join that batch, so see what you have to do when you log into Unacademy. You just need to change your course preference, right? You can select IIT JEE, and then you will get that particular batch, and you will be and you can get enrolled there, right? So that's what you need to take care. Done, students? That's what you all need to take care. So be the part of that batch as soon as possible, and do check out these videos as well. Clear, bache? So let's start this particular chapter. So in the last class, we have already discussed about the microsporogenesis. Let's start with the revision of microsporogenesis. That's what we have covered in the last class now. Your microsporogenesis, isn't it? So genesis means formation. What is the meaning of genesis? Genesis means formation. It's microsporogenesis, that is microspore formation. Isn't it? Isn't it? What is it? It is the microspore formation. So when we talk about the microsporogenesis, you know that in the case of male part, okay, in the case of male part of the flower, we know that we have the stamen. What do we have? We have the stamen, and this part here is anther, and in typical angiosperm, anther is going to be, yes bache, in typical angiosperm, anther is going to be, Rashmika, we have the Avengers batch. Okay, so in a typical angiosperm, the anther is going to be bilobed, dithecous, tetrasporangiate. Do you remember that? It is bilobed, dithecous and tetrasporangiate. That's what we know. And here we have the filament. So you know that when this anther, it is bilobed, dithecous and tetrasporangiate. So in the last class, we have even revised the structure of that microsporangium as well, right? That's how, right? We have already revised the structure of that microsporangium as well. So firstly, let's have a quick revision of that particular part. So hope you remember the layers, right? Hope you remember the layers. So the outermost layer in that microsporangium is the structure in which microspore will form. So outermost layer is going to be epidermis. Outermost layer is going to be, Trisha, epidermis. Below epidermis, what do we have? We have endothecium. Okay, below endothecium, what do we have? We have 2 to 3 middle layers. And below middle layers, there will be the tapetum. So tapetum is going to be binucleated. Actually, here the endomitosis will be there. Polyploidy we will see in the case of tapetum. Okay, so when it comes to the tapetum, it is highly nutritive and it is secretory as well, right? So it is covering this part, and here what are you going to get? This is the sporogenous tissue. Clear, bache? This is the sporogenous tissue. That's what you need to keep in your mind. Clear, bache? That's what you need to keep in your mind. Is that clear? Is that clear? So here, right, even I explained you this thing in the last class, that initially it is believed that archisporial cell is there, that will divide, will form parietal cell, will form sporogenous cell. Parietal cells are going to form these layers, epidermis, endothelium, middle layers and everything, and your sporogenous cell is going to form the sporogenous tissue. But what is concerning here, what we need to revise here mainly is, like here, when you pick up any cell, right, you know that the cells are going to be diploid here. Cells are going to be diploid here, isn't it? Isn't it? I'm talking about the sporogenous tissue. Tapetum is not diploid. Tapetum is polyploid. That's what we have covered in the last class. And guys, quickly hit the like button as well. Quickly. And if you're new to our channel, you know what you have to do. You have to subscribe our channel. So do it quickly. Do it quickly. So you know that here when you talk about the cell, fine, the sporogenous tissue. So you are going to pick up one cell. Okay. So when you talk about that cell, that is pollen mother cell or microspore mother cell. What's that? That is pollen mother cell and that is microspore mother cell. Yes, everyone. I want you people to interact with me. So quickly answer my questions in the chat section. So here what do we have? The pollen mother cell and the microspore mother cell. You know that when it's a mother cell, it will undergo meiosis. Firstly, it will be meiosis 1, which will result in the formation of 2 haploid spores. Then there will be meiosis 2. Then there will be meiosis 2, and that meiosis 2 will give us 4 haploid microspores. Clear, bache? 4 haploid microspores, and all of them are attached to each other with the help of what? Right, they all are held together with the help of polysaccharide callose. Clear, bache? With the help of polysaccharide callose. Done? Done. Are you getting my point? Right? Callose. Clear? So you know that these microspores, they can be arranged in different ways. It can be isobilateral. Okay. It can be tetrahedral. Isobilateral, tetrahedral, isobilateral in the case of monocots, tetrahedral in the case of dicots. T-shaped arrangement is also there. Decussate arrangement is also there. Linear arrangement is also there. So that's what we have discussed in the last class about the microsporogenesis. So hope this part is clear to you all. Yes. So most common type of tetrad is your tetrahedral. So I hope this part is clear to you all. Yes. So that's what we have covered in the last class, even from the NCERT. So you please have a look of this particular, right, this particular diagram as well. So this diagram is very important. The role, right, the role of tapetum is also very important. Done? The role of tapetum is also very important. Now, let's revise it from NCERT. So you know that microspores, they are arranged in the form of 4 cells. Right, there will be the cluster of 4 cells, which is the tetrad, and as I said, the most common form of tetrad is tetrahedral. Right, the most common form of tetrad is tetrahedral. Are you getting it? Yes. Are you getting it? So as anther matures and dehydrate, the microspores, they dissociate from each other and then they will develop into the pollen grains. So ultimately these haploid microspores, they will develop into what? They are going to develop into the pollen grains. Are you getting it? These, yes, these haploid, okay, these haploid microspores, they are going to develop into the pollen grains. Is it clear? Yes. Is it clear? So inside each microsporangium, several thousands of microspores or pollen grains are formed, and they are going to release with the dehiscence of the anther, and that's what we need to study next. That is your pollen grain. Clear, bache? That is your pollen grain that we need to study now. Now, as I said, you have haploid microspores. Now, bache, these microspores, they'll be covered with some layers. Right, they'll be covered with some layer. Rashmika, I think now we have we have one Avengers batch, or you can see that NEET Energized batch. Rashmika, we have closed the enrollments for that particular batch. You can check the description box if there is link for that particular batch. Be the part of that, or else I will talk to my team if it is possible to get enrolled there. Okay. Okay, so now let's focus here. So microspores. So now what is going to happen to these microspore students? There will be the layers that are going to cover this microspores. What will be there? There will be the layers. Okay, so when you talk about the pollen grain, even in plant kingdom, I told you about it. When you talk about the pollen grain, so pollen grain is your male gametophyte. What is it? It is your male gametophyte. What is the meaning of gametophyte? Gametophyte is a structure which is going to form the gametes. Okay, of course it is haploid, right? 110% it is haploid, and it is the structure which is going to form the gamete. So when you talk about the pollen grains, the outermost layer is exine, and then inner to it is intine. In the pollen grain, these are the two layers that we are going to discuss, right? The outermost is going to be the exine, and the innermost is going to be the inner one is going to be the intine. So when it is exine, right, outermost layer, do you know this exine is made up of what? This exine is made up of sporopollenin. Okay, this exine is made up of sporopollenin. I hope now you people can relate. Remember when I was teaching you tapetum, I told you that tapetum will form the Yubish bodies. What are that Yubish body? Your pre-Yubish structures, they are covered with the sporopollenin. Okay, they are covered with the sporopollenin. So when you are talking about the pollen grain, which is the male gametophyte, it is having two layers. The outer one is the exine, and the inner one is going to be the entine. And entine is made up of pectocellulose. Right, cellulose and pectin, it is making the entine, and when it comes to the exine, it is the sporopollenin. These are the layers of the pollen grain that you people should know. Okay, these are the layers of the pollen grain that you all should know. Clear, bache? Clear, bache? Right, so structure of pollen grain we need to discuss first, and then we will talk about its viability and everything. Okay, okay. So when it comes to the pollen grains, right, if I have to talk about its diameter, so pollen grain is 25 to 50 micrometer in diameter. It is 25 to 50 micrometer in diameter. Are you getting my point? Are you getting my point? And here if I talk about the exine, this exine is having, this exine is made up of sporopollenin. Sporopollenin is the strongest organic substance. We are going to talk about it. And moreover, Akhilesh Bache, when it comes to this exine, it is having different colors and the shapes as well. Okay, it is having different colors and the shapes as well. Okay, so pollen grains, you know that spherical 20 to 50 micrometers in diameter, bache. Outer wall is made up of is exine, inner wall is made up of, yes, inner wall is made up of pectocellulose. Clear, bache? Clear, bache? Yes. So now what you have to write down? You have to write down about the sporopollenin. So sporopollenin is the strongest, it is the strongest organic substance. Okay. And yes, you have to focus here, bache. It is organic, not inorganic. Okay, it is organic, not inorganic. So it is the strongest organic substance, and let me tell you, it is non-degradable. Right, we cannot degrade it. Right, you can say that strong alkali, the enzymes, the high temperature cannot destroy this exine. They cannot destroy this exine. Why? Because exine is made up of sporopollenin. Right? So it is the strongest organic substance that you know. What is it? It is the strongest organic substance that you know. And it is, as I said, it is non-degradable. Right, bache? It is a non-degradable. Clear, bache? What is it? It is non-degradable. No alkali, strong acid can destroy it. Moreover, the sporopollenin can withstand high temperature. Okay, it can withstand high temperature. No enzyme discovered that can destroy it. Clear, bache? Clear, bache? So neither alkalis nor acids, even the high temperature cannot destroy this sporopollenin. It is that strong, and no enzyme discovered so far can destroy it. Okay, can degrade it. So you know that tapetum is basically, tapetum is going to release the sporopollenin. Clear, bache? It is the tapeworm that is going to release the sporopollen. So remember, so exine is made up of the sporopollen, so it will not be degraded so easily. It will not be degraded rather. Clear? Clear. And entine, as I said, it is made up of pactocellulose. Now, what you need to understand here that, ma'am, okay, fine. Pollen grain is the male gametophyte. It will form the male gametes. And if exine is that strong, right, then how that male gametes will come out from that pollen grain? Now, this is the next question, isn't it? That if exine is that strong, then how that pollen grains are going to, right, how that male gametes are going to come out of it? First thing is that, right? So that's what now we have to discuss. So here the most important thing is layers, and you should know that exine is made up of sporopollen, and about sporopollen you need to know. That's important. Clear, bache? That's important. Now the next part is, see, so here when you talk about this sporopollen, right, because we know that it cannot be degraded, it cannot be degraded, but we have to send the male gametes out of this pollen grain. Then what is going to happen, bache? At places when you talk about this exine, it is no doubt this exine is thick, it is rigid, right? It is thick, it is a rigid layer, but it is not continuous. Okay, it is not a complete layer, it is not continuous. Do you know that? Yes, do you know that this particular layer, that is your exine, right, it is not continuous? This is what you need to remember. Okay, this is what you need to remember. It is a discontinuous layer. Are you getting it? Are you getting it? Yes, this is not a complete layer. Like at places you will see, in the case of exine, at places you will see, like let's say here, that this exine is thin. It is not thick at places, it is thin. Actually here you are going to find one structure which is known as a germ pore, right? What will you see here? You will see the germ pore. So exine thick and rigid, but at places it is thin, right? It is thin. So where it is thin now, the sporopollen is absent there, right? What is absent there? The sporopollen, right? The sporopollen is absent there. So you are going to see the germ pore there. So in the case of dicots, right, we see 3 germ pores. Mostly you are going to see 3 germ pores. And in the case of monopods, right, there will be 1 germ pore. So germ pore is an area in the exine which is quite thin. Here sporopollen is not there. And what is the purpose of this germ pore? From this germ pore, the pollen tube is going to come out. Are you getting my point? The pollen tube, right? The pollen tube is going to come out, and with the help of that pollen tube, the male gametes, right? The male gametes will come down. So dicots are having 3 germ pores, monocots are having 1 germ pore, 3 germ pore. Tricolpate is the word here. Because in dicots 3 germ pores are there. So which term are we going to use? It's tricolpate, right? And when it is the monocot, the term is going to be monocolpate. Clear, bache? What is it? It is going to be monocolpate. So tricolpate, monocolpate, tricolpate, monocolpate. Vijay, please join the Telegram group for that All India mock test series, right? You will get the test syllabus there. Okay. Clear? So that's what you have to remember. And do you know what? Because of this exine, right, because of this exine, it is made up of sporopollen, which will not be which is non-biodegradable, right? Because of this exine pollen grains, you know, they remain stored, right? They become fossilized. Are you getting it? Right, they are well preserved as fossil because of the presence of the sporopollen, right? They are well preserved as fossils because of this sporopollen. Are you getting it? Done? So that's important. Please, right, please revise it. And as I said, the entine, it is going to be thin layer, it is continuous and made up of pactocellulose. Okay, it is made up of pactocellulose. Clear? Okay, so germ pores, they are very important. Okay, done bache? So if there is any doubt, do let me know. If there is any doubt, do let me know. Clear? So, and you know what, this exine is having, you know, different patterns as well. I told you, it is ornamental as well. So this exine is having different arrays, different patterns, and it is used in taxonomy, right? You know, taxonomy. So so basically, when we need to, right, when we need to put plants into different groups, or you can say that when we need to classify the plants, right, bache? So we can even study the pollen grains. The study of pollen grains help in that taxonomy. The study of pollen grains help us to, you know, put that, to classify that particular plants. Okay, so this exine is very useful in that case. Fine. So it is having different arrays, patterns. Okay, and obviously it is going to be species specific as well. And as I said, the exine is what? It is ornamental as well. It is having different colors and all. Okay, so that's what you have to remember. So this is the important part related to the pollen grains. Do revise it, right? You should know these terms, monocolpate and the tricolpate as well. Okay, right, and the tricolpate as well. Okay, done. So now bache, you know, jaise pollination is what we need to, you know, that here in the case of plants, your male gamete is not going to be motile. It is not at all motile. So with the help of some agents, the pollen grain, we need to transfer the pollen grains to the stigma of the female part, isn't it? We need to transfer the pollen grains to the stigma of the female part, to the stigma of the pistil, right? That is what pollination is. The transfer of pollen grain is what? Pollination, right? So when we use the insects for pollination, that is entomophily. When we use the insects for pollination, that is entomophily, right? Insects are helping in that pollination, right? And you know that honeybees are very common in that case. So what is going to happen? These pollen grains, because these honeybees, they will visit flowers, okay, for the nectar, of course. So if pollen grains will be sticky, they are going to join to that honeybee, isn't it? And whenever honeybee will move from one plant, one flower to the another flower of same species, obviously that pollen grains, right, they will leave that pollen grains on that other flower, of course, right? So in this way the pollination will occur. So in that case, especially when, you know, a flower is dependent and when a plant is dependent on this insect pollination, in that case what is going to happen? Your pollen grains are covered with a sticky layer, right? Pollen grains are quite sticky. Okay, right? Why are they sticky? Why are they are having that oily secretions, right? So that they can join to the insect body. So actually the pollen kit formation will be there. What is going to happen in that case? The pollen kit formation is there. Okay, okay, so you will read this word pollen kit. So pollen kit is nothing, it is just the group of pollen grains. Obviously some oily and sticky secretions are going to hold them together. Done bache? They are going to hold them together. Is that clear? Yes or no? Is that clear? Okay, so that oily layer around the pollen grain is pollen kit. And when you talk about the pollen kit is having that lipids, carotenoids, right? It is having that lipids, carotenoids. Done bache? So that's about the pollen grain. That's about the pollen grain. And as I said, pollen grain is male gametophyte. It is going to form the male gametes. Okay, so can you tell me about the female gametophyte? Yes. Can you tell me about the female gametophyte? Pollen grain is male gametophyte, but can you tell me about the female gametophyte? Very good, very good. Done bache? Okay. Sure. So that's about the pollen grains. Now let's revise it from the NCERT. So here you can see that they represent the male gametophyte. So kindly highlight this part, right? Kindly highlight this particular part, that pollen grains, they are representing the male gametophyte, right? So see, they have given the example that if you touch the open anthers of hibiscus or any other flower, you might find deposition of yellowish powdery pollen grains on your finger. And in the case of hibiscus, it's very common that yellow powder that we feel on our fingers, isn't it? Isn't it? So sprinkle these grains on a drop of water taken on a glass slide, and you are going to observe, right? You will observe these pollen grains basically. So here you can see scanning electron micrographs of a few pollen grains. So just look at this exine, Hannah. It is having different patterns, it is having different arrays. Okay, so you can see even the shape, it varies. So their layer, this exine is having different patterns. Okay, this exine is having different patterns. Is that clear? Right? Is that clear? So after this, the next topic that we need to study is the development of male gametophyte, right? This diagram, it is basically under the development of male gametophyte. We will talk about it. But yes, let's revise the NCERT first. Okay, let's revise this particular part from the NCERT first. Okay, so you can see here, you will really be amazed at the variety of arc sizes, the shapes, the colors, and the designs seen on the pollen grains. So in different species, this shape, design, patterns, they are different in the pollen grains. Clear bache? Clear bache? So as I said, they are generally spherical. What are they? They are generally spherical, right? 25 to 50 micrometers in diameter. 25 to 50 micrometers in diameters. And as I said, they have two prominent walls. So outer layer is the exine, right, which is made up of sporopollenin, which is the most resistant organic material. So this word is very important. It is resistant, but it is organic. It is not inorganic, it is organic. That's what you need to focus. Okay, that's what you need to focus. Clear? So it can withstand high temperature and strong acids and alkalis, and no enzyme that can degrade sporopollenin is so far known. Okay, so it is having prominent aperture. Exine is having prominent aperture. That is your germ pore. Okay, so at the place of that germ pore, sporopollenin is absent. Your exine is very thin there because sporopollenin is absent there. So exine is not a complete layer. It is not a continuous layer, but your entine is a continuous layer. Clear? But your entine is a continuous layer. Clear, bache? So because of sporopollenin, pollen grains can be preserved as fossils as well. Okay. Done, bache? So inner wall of pollen grain, as I said, it is going to be thin, continuous, and even it is elastic, bache. It is having pectin and cellulose, that is your pectocellulose, and as I said, it is elastic as well, your entine. Done, bache? Your entine. So the cytoplasm is obviously surrounded by plasma membrane. 110% plasma membrane will be there, then entine, then exine will be there. Clear, bache? Then entine, then exine will be there. Done? So now we will talk about the development of male gametophyte, that is development of your pollen grain. Okay, development of your pollen grain. So mature pollen grain. Okay, mature pollen grain is two-celled. What is it? A mature pollen grain is a two-celled stage. Is having two cells, or it is two-celled. That's what you have to write. Mature pollen grain is two-celled. Now what is the meaning of this? Actually, what is going to happen in this microspore or in this pollen grain, we have the haploid nucleus, isn't it? We have the haploid nucleus. Now bache, it will undergo mitosis. Which division will be there? Mitosis. I hope you remember this part, that meiosis will only take place in the diploid cells. Right, Subhashini? Meiosis will only be there in the diploid cells. Right? So here in the pollen grain, mitosis will occur. You know that what mitosis is? Mitosis is the equational division. Now what is the meaning of equational division? The division in which parent cell and the daughter cells, they have same number of chromosomes. What is the meaning of equational division? The division after which the daughter cells are also having same number of chromosomes like the parent cell, right? Like the parent cell. Clear, bache? Clear बच्चे? Nikhil, Vijay, please do not spam. You will get your syllabus in the Telegram group only. Okay? I cannot provide you the test syllabus here. You will get it in your Telegram group. So kindly have patience and please focus here. Done. So equational division will be there. But here, this mitosis is unequal. What is it? It is unequal. Actually, the spindle apparatus that is going to form here, it is asymmetrical. Okay, it is asymmetrical. So after mitosis, what is happening? No doubt you are going to get two haploid nuclei. No doubt you are going to get two haploid nuclei. But as I said, asymmetrical mitotic spindle is there. So the two cells that you are going to get in the pollen grain, they are having, right, they are having unequal sizes. Their sizes are not equal. Okay, the small one, right, this small one, it is spindle shaped. It is spindle shaped and it will be there in the cytoplasm of the larger one. Okay, so this larger pollen grain, it is what? It is basically the vegetative cell. It is basically the vegetative cell, also known as tube cell. It is vegetative cell, also known as tube cell. What is it? It is the vegetative cell, also known as tube cell. And the smaller one is what? It is the spindle shaped. What is it? It is the spindle shaped, and this smaller cell is what? It is known as bache generative cell. Okay, it is the generative cell. So if I'm saying mature pollen grain is two celled, so which two cells are there? One is your vegetative cell. Of course, it is also haploid. Okay, and another is your generative cell. Another is your generative cell. So vegetative cell is also known as tube cell. What is it? It is also known as tube cell. And this generative cell is the one which will form male gametes. Okay, it is the one which will form male gametes. And bache, the vegetative cell, the tube cell, obviously it is going to form the pollen tube. Fine, it will form pollen tube. Is that clear? Is that clear? So that is basically the development of male gametophyte, development of the pollen grain. That this pollen grain, right, the mitosis will occur, unequal spindle apparatus will form, due to which the two cells, they are having unequal sizes. The smaller one is spindle shaped, which is going to float here in the cytoplasm of your larger cell. The larger cell is vegetative cell, also known as tube cell, and it is the one which will form the pollen tube. Okay, it is the one which is going to form the, yes bache Karthinga, it is going to form the pollen tube. So that's what you have to remember. Clear bache? That's what you have to remember. Now in the NCERT there is one point, like in 60%, in 60% of angiospermic families, pollen grain, when it will be dispersed, right, when there will be the dehiscence of the anther, the pollen grains that are released, right, in 60% of the angiospermic families, bache, they are having these two cells. Okay, they are having these two cells. Are you getting my point? What do they have? They are having these two cells. Are you getting it? Yes or no? Yes or no? Right, in 60% of the angiospermic families, pollen grains are released in two-celled stage. Fine. And in remaining 40%, it will be in three-celled stage. Now, what is the meaning of this particular statement? What is the meaning of this particular statement? See, when it is two-celled stage, there will be one generative cell, okay, plus there will be the vegetative cell. What will be there? There will be the vegetative cell. And when it is the three-celled stage, no doubt, here vegetative cell plus two male gametes. What will be there? There will be the two male gametes. So now the topic that we are discussing, it is what? It is the development of male gametophyte. That's what we are discussing. It is actually the microgametogenesis, that how that microgamete is going to form. So it is actually the microgametogenesis. Clear bache? Clear bache? Done? Done. So actually all this process, where is it taking place? See, let's discuss it here, okay? You know that this is the pollen sac, isn't it? And here in between these pollen sacs, you know that the parenchymatous cells, they are present, right? This tissue part is present, okay? That's what we know. Now, as you know that this development is going on here within this pollen sac. So ultimately, can I not say that when you talk about the pollen grains, their development, yes, their development will be there, right, in this pollen sac. So basically it is taking place because the development is taking place inside the pollen sac. It is basically at mother place where development is going on, है ना? When you talk about the development of pollen grains, it is at mother's place. Mother's place means to that particular flower, in that particular flower the development is going on, right? It's not like that pollen grains are growing or they are germinating on some another flower. No, it's not like that, right? Wherever they are forming, they will start their development there only, right? They will start their development there only. Okay, in situ development, you can say that. In situ development, that development is there at the mother's place only, isn't it? Development is there at the mother's place only, right? Pollen grains, they didn't leave the mother's place, but they start their development. They have started their development there. That's the point here. Okay, so it is taking place at mother's place, kind of in situ development is there. Done, bache? Done, bache? That's the first point. So no doubt this development will be there, and you are going to get two-celled stage in 60% of the angiospermic families. What are you going to get there? You are going to get two-celled stage in the 60% of the angiospermic families. That is, your pollen grain, the haploid nucleus will divide. You will get two unequal sized cells, larger one and the smaller one. This is the two-celled stage. And you know that where that process is taking place? Within this, yes, within this pollen, yes, within this pollen sac. Is that clear? Within this pollen sac. Is that clear? Yes or no? Is that clear? That's important as well. So I hope it is clear to you all. Okay. That's what you need to keep in your mind. Okay? So that is happening here in the pollen grain. Now, you know which two cells are there. So after this, right, once these pollen grains, they will be released. Let's say this pollen grain is releasing in its two-celled state. See, in most of the families, what is the scenario? In most of the families, what is the scenario? They are going to pay for your fee. है ना? They will be like, ठीक है, now you are the graduate. Or some families till your post graduation, they are like, ठीक है, we are giving you the money, okay, you want to study, you can, but then you have to earn, right? In majority of families, that's the scenario. They are paying our fee, and then they are expecting that after a certain age that we will start earning, isn't it? We will start earning, isn't it? That's what, right? That's the reality, isn't it? That's the reality. And now in some families, when they have a lot of money, they over-pamper their kids. So they are like, it's okay, baby. No need to get stressed. We are going to give you the money. Okay? We are going to give you the money. There is nothing, right? You have no need to start something by your own. You have no need to do for any struggle or something, right? We are going to pay for your expenses. Don't worry. So in 40% of the angiospermic families, that's the scenario. So in 60% of the families, when pollen grain will be released in two-celled stage, you know that where that pollen grain will go. Obviously, its landing platform is the stigma. Understand this process first, bache, then we will revise it from NCERT. Its landing platform is what, Pinky? It is stigma, right? So if this pollen grain, right, and this stigma, if they are compatible, if they are from same species, if they have the compatibility, then obviously the pollen tube formation will be there. What is going to happen? The pollen tube, Pinky, the pollen tube formation will be there. And what is going to happen? Your vegetative cell, as I said, your vegetative cell, your tube cell is going to form the, it will form the pollen tube, and that generative cell, it will again undergo mitosis, and it will form two male gametes. So basically in these, in this pollen tube, you will get three nucleus, right? One is of that vegetative cell, and two male gametes will be there and their nuclei. Are you getting it? Are you getting it? So in 60% of the angiospermic families, this is the scenario that if pollen grain, it lands on the compatible stigma, it will grow, it will form the pollen tube. Now in this pollen tube, you will be having that nucleus of that tube cell, plus you are going to get two male gametes. How that male gametes will form? How that male gametes will form, bache? By the mitotic division of your generative cell. So your generative cell, it is already haploid, so of course it will undergo mitosis and then it will form two male gametes. So ultimately male gametes form within the pollen grains. So that's why we say that pollen grain is the male gametophyte. So pollen grain is not the male gamete. Pollen grain is male gametophyte. What is the meaning of gametophyte? The structure that will form the gametes. Right? The structure that will form the gametes. Is it clear? Is it clear? Clear? And in 40% of the angiospermic family, this pollen grain, right? This pollen grain will grow at mother's place. You will get vegetative cell as well, and you will get two male gametes there. So obviously in totality 3 cells will be there. 3 cells will be there. Done. So that's the meaning of this and this is basically the development of male gametophyte, or you can also call it as microgametogenesis. So microgametogenesis means formation, microgamete means microgamete formation. Clear? Clear? So firstly we will start the microsporogenesis, that how microspores will form. Then we talk about this development of male gametophyte. And in this male gametophyte, you know that your microgametogenesis will occur. Is that clear? Yes or no? Is that clear? Sure. Now, in this diagram, so ultimately, this process is taking place here. So now, when you talk about a mature anther, do you know in that mature anther, you will only get epidermis and endothecium? Are you aware of it? That in mature anther, you are going to get these 2 layers only, epidermis and endothecium. You are not going to get the middle layer here. Yes, are you aware of it? In the outer coverings, only these 2 layers are there. You are not going to find the middle layers here. Why? Because you know that from the middle layers, all the food will be taken and it will be given to the developing pollen mother cells or to the developing pollen grains or the developing microspores. Okay. So when it comes to the outer coverings, outer covering of this mature anther will contain epidermis and endothecium only. Now, you know that because it is growing in pollen sac, you know that pollen grains are forming. Now, can you see in between these 2 microsporangium, here you have some other tissue as well made up of parenchymatous cells. Can you see this? Here actually what is present? The parenchyma cells. Parenchymatous tissue is there. So bache, in mature anther, you know what is going to happen? Even this sterile tissue, right? It will not be there. Actually both of these pollen sacs, they will get fused. Okay, they are going to get fused. Are you getting it? Are you getting it? Yes or no? Yes or no? So basically the tissue present here in between these 2 pollen sacs, it will be degenerated. Your pollen sacs, they will fuse. Okay, they will fuse. So when you look at this particular part, when you look at the mature anther, you will see it will contain only, right? It is not having 4 microsporangia. It will not contain these. Each lobe is not having 2 theca. You can say that 1 theca will be there. But now obviously it is not the theca word that we are going to use. Okay, because they are mature. So now you know that it's not theca. Theca will grow, microsporangium, then pollen sac is there. So in mature pollen grain, in one lobe, you will see only 1 pollen sac. Another lobe, you will see another pollen sac. So ultimately they will fuse, their sterile tissue will be degenerated. This point clear? This point clear? Even in the last class, I told you that this part is the stomium, right? And here you are not going to see the callose thickening and alpha cellulose thickenings in the endothelial cells. I told you last time, right, about the stomium, right? So basically, bache, in the case of, in majority of angiosperms, this longitudinal groove, right? the dehiscence will occur from that longitudinal groove, right? It is that longitudinal groove from where the dehiscence of anther will occur. Are you getting my point? The dehiscence of anther will occur. You can see it here. Then longitudinally that will occur. And when will it takes place? It takes place in the dry season. Why in dry season? Why in dry season? Actually, in dry season, what is going to happen? From endothelial cell, all the water, right? Endothelium is hygroscopic in nature. That's what I told you. Endothelium is hygroscopic in nature. That's what I told you. But when it is the dry season, in the endothelium, the water will not be there. The water will not be there, right? Right? So now you know that the radial and the inner wall is having the cellulose, alpha cellulose thickenings, right? Because water is lost. Now these cells, they will change their shape. The detail is not required. I'll tell you what is the most important point here. So imagine a layer where inner side and the radial walls are having alpha cellulose thickenings. It's a dry season. Water is not present then. Okay, it's a dry season. Water is not present then water will be out. Water is out. So it will change the shape of that cells. That cells, because of that thickening, they are not losing their shape, right? They will try to maintain that shape, but it is not possible because water is not there. So some curves will appear in the cells, right? It's the cells, the layer will become curved. So actually that whole thing is going to exert a pressure, right? It is going to exert a pressure, and from the stomium, right, there will be the dehiscence of the mature anther. Clear, bache? There will be the dehiscence of the mature anther, right? So ultimately, walls of your endothelial cells, they will contract because they do not have the water, right? But because of alpha cellulose thickenings, the radial walls and the inner walls, they will not contract, right? The curves will appear in the layer, and basically a force will be generated over the anther, and because of that, the stomial cells, they will break. Clear? The stomial cells, they will break. Done. So longitudinally dehiscence is there in the majority of anther. So as I said, in mature anther, you will see only one pollen sac here, because two pollen sacs, they are fused, only one pollen sac here, one pollen sac here. So that's how it works. Clear, bache? That's how it works. So I hope even this diagram is clear to you, that what is actually happening here. Okay. So now let's revise this part from the NCERT, and then we will start the female part. So you guys can see the diagram here. Okay, so vacuoles are present here. This is the nucleus. It will undergo the mitosis. Asymmetrical spindle is there, and you will get these two cells. Fine, you are going to get these two cells. So when the pollen grain is mature, it contains two cells, the vegetative cell and the generative cell. Okay, the vegetative cell and the generative cell. So vegetative cell is bigger, has abundant food reserve, and a large irregularly shaped nucleus. That's what you need to remember. Vegetative cell is having the food reserve as well, right? It is having the food reserve as well. Clear? And the generative cell, which is small, spindle shaped, it is going to float there in the cytoplasm of the vegetative cell. Clear? Clear, bache? So small cell is spindle shaped with dense cytoplasm and a nucleus. Clear? So in 60% of the angiospermic families, as I said, the pollen grains, they will shed at a 2-cell stage. They are going to shed at a 2-cell stage, and in remaining species, it will be the 3-cell stage. Done, bache? Clear? So if there is any doubt, do let me know. Any doubt? Sure? Okay, so the next part here is that pollen grains of many species, they cause severe allergy. Now, things about the pollen grains, you know that some people are allergic to that pollen dust. When there is the pollination, right, during that particular season, they start sneezing and all. Some people are even having the asthma. So pollen allergy is very, very, very common. Okay, pollen allergy is very common. So pollen grains can, of many species, they can cause severe allergies and bronchial afflictions, right? So they can have respiratory disorders like asthma, bronchitis can be there. Fine. Done, bache? So jैसे parthenium is what? It is known as carrot grass. What is it? It is known as carrot grass. You all have seen this weed. Even you can Google it after the class. Okay, parthenium, it's a very common weed. So its pollen grains are quite allergic. Done, bache? So it is actually the invasive species. Do you know that? right? It is actually the intruder species. It is not from India. See, it is written here that it came into India as a contaminant with imported wheat and has become ubiquitous in occurrence and cause pollen allergy, right? So from some other country with the imported wheat, it came here in India and then it got widely distributed. Fine. Fine, bache. So pollen grains, they are also rich in nutrients. Okay, so in Western countries, it has become fashion that they take pollen grains as food supplements, right? Pollen tablets as food supplements, right, bache? So large number of pollen products are there in Western countries, like the tablets, the syrups. They are available in the market, and their consumption has been claimed to increase the performance of athletes and racehorses. Done. So this is about the importance of pollen grains, that how can we use it other than this pollination and all. Clear? So these are the pollen products, bache. Done. So now next topic that we need to discuss is viability of the pollen grains. Fine. What we need to discuss? Viability of the pollen grains. Viability means their capability to germinate. What is this viability of these pollen grains? Their capacity to germinate. Okay. Right, it is not the case that always pollen grains, like let's say today, right, pollen grains, they shed off from their parent plant and then they will remain viable for years. It is not the case. It varies from species to species. Okay, their viability, it varies from species to species. Like see, when once they are shed, they have to land on the stigma before they lose viability, if they have to bring about the fertilization. Of course. Done, bache? So period for which pollen grains remain viable is highly variable and to some extent depends upon the prevailing temperature and humidity. Right. Species to species, no doubt this viability, it varies, but external factors are also playing the role, like the temperature, the humidity. Okay, like the temperature and the humidity, it is also playing the role. Like in the case of rice and wheat and pollen, in the case of cereals such as rice and wheat, pollen grains, they lose their viability within 30 minutes. This is important. In the case of rice, in the case of wheat, pollen grains, they are going to lose their viability within 30 minutes of their release. It is important. Please note down in your notes, right? But in some members of Rosaceae family, of Leguminosae family and Solanaceae family, they maintain viability for months. Like in some species, they can be viable for minutes. In some species, they can be viable for months, right? And in some species, they can be viable for years as well. It is also the case. Okay, so that's what you need to remember. Like in the case of rice and wheat, within 30 minutes, in the members of family Rosaceae, Leguminosae and Solanaceae, they'll be viable for months. So now what's your homework? In the comment section, you will write down the examples of Rosaceae, Leguminosae and Solanaceae and the examples that you have studied in the chapter Morphology of the Flowering Plants. Clear, bache? Clear, bache? And do you know that these pollen grains, we can even store them? This is a kind of ex situ conservation. This is what we are going to study in the biodiversity, right? Like let's say if any plant species or even the animal species, if they are under threat, if there are chances that they will become extinct in the near future, we can even conserve them. How? There are two ways, in situ, ex situ, right? There are two ways. In situ mode is also there, ex situ mode is also there. When we use the term in situ, okay, in situ means on site. Like let's say a plant is growing in a particular area. Okay, that plant is growing in a particular area, we will make sure, right, that it will stay protected there. Okay, on site, wherever they are living, wherever they are present naturally, in their natural habitat, we are going to provide them the protection. In the case of plant as well, in the case of animal as well. But when it is ex situ conservation, it is off-site conservation, means they are not safe in their natural habitat. So what we have to do, we have to protect them. We have to take them to the special habitats. Okay, bache, where all that protection, food, everything will be provided to them. So when you talk about the ex situ conservation, the most advanced way here is cryopreservation. The most advanced way is what? It is the cryopreservation. What is it? What is it? It is the cryopreservation. Are you getting it? So when you use the word cryopreservation, what is going to happen? The pollen grains, even the semen, the sperms, in the case of animals, it will be conserved. How? At minus 196 degrees Celsius in liquid nitrogen. How are we going to conserve it? In liquid nitrogen at a temperature of minus 196 degrees Celsius. So what is it? It is an advanced way of XC2 conservation, and we used to call it as cryopreservation. What is it? It is the cryopreservation. Are you getting it? Yes, bache. It is the cryopreservation. So such pollen grains, they'll be stored, like let's say one plant species is, might be that plant will become extinct in near future. So in that case, we can store the pollen grains of that particular plant. We can make the pollen banks. Okay, we can later use them for the crop breeding programs. Done. So they will be conserved here. So cryopreservation is a mode of XC2 conservation, and it is the most advanced way of XC2 conservation. Cryopreservation it is. Done, bache? Cryopreservation it is. Done. So the next topic that we need to start is the female part. So the pistil or the carpel. Fine. The next that we need to start is what? It is the pistil or the carpel. Done. Gynoecium. That's what we need to discuss next. What is it? It is the gynoecium. Done, bache? It is the gynoecium. Okay. Any doubt? Done? So you know that carpels, right? Group of carpels, they are going to form the gynoecium. What is this gynoecium? It is the female part. What is the gynoecium? You know that it is the innermost whorl of a flower. Gynoecium, it is the innermost whorl of flower, and it is the female part actually. What's that? It is the female part. Done. So now when you talk about the gynoecium, you are going to talk about what? You will talk about the pistil or the carpel. That's what we are going to discuss here. Pistil or carpel. So this pistil or carpel is having three parts: stigma, style, ovary. It's stigma, style, ovary. So you can see here this part, the landing platform for the pollen grain, stigma. This tube-like structure is style, and this swollen part of the pistil or carpel is the ovary. Clear? The swollen part is the ovary. And now, bache, in the ovary, the space here, it is known as ovarian cavity or locule. What's that? It is ovarian cavity or the locule. And here, what will you find? You will find the ovule. Clear, bache? So this is the ovary, the swollen part of this pistil, right, bache? Here the space within this ovary is known as ovarian cavity or ovarian locule. Ovarian cavity or ovarian locule. And here in this ovarian cavity or locule, you are going to find what? You are going to find the ovule. So I hope in morphology of flowering plant, that's what I hope you have studied that. Right? It's not mandatory that this ovary will be unilocular. See, we will use the term unilocular when there is only one ovarian cavity. If there will be two ovarian cavity or more than two, I'll give you one example of axile placentation. Do you remember axile placentation? In placentation, we will see that how these ovules, they are arranged, right? How are they arranged there? So I hope you know about the axile placentation. We have studied that in the morphology of flowering plants. Axile placentation is there in your tomato. It is there in your citrus. It is there in your orange. Like basically in citrus fruit, it is present. In family Rutaceae, we see the axile placentation. So you are even going to see that the ovary, right? It will be multilocular or it will be trilocular. Like here, it is unilocular if there is only one ovarian cavity. If there is more than one, like let's say 3 ovarian cavities are there, it will be trilocular. If like in axile placentation, that's what we discussed. We see that ovary, it is trilocular or multilocular. That's what we read there. Okay. Like I'll give you one example. In oranges, you know that So many seeds are there. So many seeds are there. So many seeds means, and even in lemon, you know that when you cut it from the center, when you take its transverse section, so what will you see? So these things are what? They are the locules, and you know that you are going to see the seeds here, right? So means so many ovules are there, so many locules are there. So that's how it is arranged. That's how it is arranged in the ovary. So ovary can be unilocular, it can be bilocular, it can be trilocular, it can be multilocular, right? And the ovules will be present here, and these ovules after fertilization, what will they do? What will they do? They will form the seed. These ovules after fertilization, what are they going to do? They are going to do, right? They are going to form the seeds. Clear, bache? Clear, bache? Right? So that's what we need to remember. Usually in one locule we see one ovule, right? That's the case usually, that in one locule you're going to see one ovule only. Done, bache? That's what you need to remember. So carpel, pistil, remember it is equivalent to, in last class also I told you about this, it is equivalent to megasporophyll. Like your stamen is equivalent to microsporophyll, same way this carpel is equivalent to megasporophyll. This carpel is equivalent to megasporophyll. Are you getting my point? Done, bache? So free end is basically your stigma, the swollen end, the basal part of the pistil is the ovary. So if there is any doubt, do let me know. Done? Sure? Sure. So now the next part that we need to understand is, see, yes, a flower can be, it can be monocarpellary, or you can say that gynoecium can be monocarpellary, it can be multi or polycarpellary. It's not mandatory, bache, that always there will be single carpel in a flower. No. Right? No, of course not. See, mono means single carpel, right? And when it is multicarpal or polycarpellary, means there are many carpels here. Okay, so flower can be monocarpellary, that is only one carpel is there. It can be multicarpellary, that is many carpels are there, right? Multicarpellary means many carpels are there. Now if there are more than one carpels, so obviously there can be two fates, isn't it? Let's say you are living in a family where only one person is there, then of course there will be no fight. That single person is not going to fight with anyone else, right? But if there will be so many family members, so two possibilities are there, right? Either they will fight or they will live all together. Either there will be love or there will be the hate. That's the scenario, right? That's how you can understand it. So when you are talking about the flower is monocarpellary, it can be multicarpellary. And if it is multicarpellary, two possibilities can be there, right? Your gynoecium can be apocarpus, it can be syncarpus. Okay, it can be apocarpus, it can be syncarpus. So when the word is apo, A, that's my way to remember this, A for alone. Like let's say in your family, so many members are there, but they are not in love with each other, right? Right, they all are living individually. Although they are the part of same family, but they have no interaction. They are all alone, but living in a same family. So that's how I remember it. So multicarpellary, apocarpus. Apocarpus means all the carpels, they are free, they are not fused. They are living individually, they are not free, they are free, they are not fused. Okay. So here you guys can see that free pistil or free carpels are there. Free carpels. Now syn. In general, you know that what is the meaning of syn? Syn means fused. So all that carpels, they are fused. So it is possible only in the case of multicarpellary ovary, of course. It is possible only in the case of multicarpellary ovary. Isn't it? It is the possibility there only. Apocarpus alone, right? Syncarpus fused, they are fused. So the examples we need to remember. So what is the example? One is your ma'am Another is your PMS. Ma'am, PMS. Clear? Ma'am, PMS. That's the example. So, ma'am, M stands for multicarpillary. M stands for multicarpillary. A stands for apocarpus. M stands for mycelia. Right? This is the example which is given in your class 12th NCERT. That mycelia is the example in which multicarpillary ovary is there and it is apocarpus. Now, when it is PMS, P stands for papaver. P stands for papaver. M stands for again multicarpillary. Right? It stands for multicarpillary. And then it stands for syncarpus. S stands for syncarpus. So these are the examples which are given in your NCERT. So mycelia and papaver. Mycelia and papaver. But bache, you can also add 2 more examples which are given in your class 11th morphology of flowering plant chapter. So here you have rose and the lotus as well. And here, other than this papaver, you have the example of hibiscus as well. So they are also the part of NCERT, but they are given in, yes bache, they are given in your class 11th book, right? They are given in your class 11th NCERT. So is it clear? Yes. Is it clear? Sure? Sure. So that's what you need to remember. Okay. So see, gynoecium represents the female reproductive part. It is having, if single pistil is there, monocarpillary. If more than one pistil, multicarpillary. So when there are more than one pistil, if they are fused together, syncarpus. If they are free, apocarpus. See, you should know these words which are written in bold. Okay, which are bold basically. The stigma, style, ovary, the 3 parts of your pistil. Stigma is the lending platform, right? Style is the elongated slender part. And when it comes to the ovary, the basal bulged part. Okay, so inside ovary, I told you already, ovarian cavity or locule is there. The placenta is located inside ovarian cavity, and to this placenta, your ovules are attached. So this is the diagram that you need to remember. Clear? So it's the dissected flower of hibiscus. Fine. Hibiscus. Hibiscus, your China rose. So, bache, that's your homework as well. Right. After this class, you will open up your morphology of flowering plants, and wherever China rose is given. Right, just say in actinomorphic, zygomorphic, wherever it is given, that's your headache. You will write it down in the comment section. Will you do it? Yes, Ramesh. Will you do it? In the comment section, this is what you have to put, that ma'am, China rose in the chapter morphology of flowering plants. It is the part of this. It is the example of this. That's what you have to do. Are you going to do it? Tell me. Are you going to do it? And trust me, in this way, you can make your notes better. Paka, promise, take it down. So next, here you can see syncarpus, papaver. Isn't it? Papaver. And this one is your mycelia. And that is your ovule. So ovule we will start in the next class. And let me tell you, it is the ovule which is the megasporangium. Okay, it is the ovule which is the megasporangium. Clear? Clear? It is the ovule which is the megasporangium. So this part we will start in the next class. Okay. So tomorrow your class is there, but it will be, the class is at 11 AM. Done, and we will finish this chapter. In that class, what will we do? We will finish this chapter. Done? Sure. And students, let me tell you this thing. See, if you are sure that yes, you have covered the syllabus, if you are taking any coaching classes also, and if you are studying by your own, you should give the, right, you should give the test, you should give the mock test, and you know that for that you can join our All India mock test series. And now, like some students are asking for the syllabus, we have a separate Telegram group for that particular test series, and in the Telegram group you will get the syllabus for the upcoming test as well, and we'll make sure that you will get it today. Okay. Okay, so join that test series, start practicing the question. Every Monday you used to have test discussion sessions as well, in which we take your doubts and we, you know, practice the questions again. So please join it. And see here, again I'm telling you, this is the video that I have made, right? In this video, clear? In this video, I have explained that what exactly is this new NCERT, what is the role of that QR code, how you have to use that QR code. And if you have any confusion related to NT-NEET bill, it is given here, right? The mind maps are provided to you, and for that mind maps, right, if you want to revise your entire chemistry with the help of tricks, with the help of mind maps, with the help of best explanations, then you have to join the batch of HSP Search batch. And let me tell you, this batch is one of the best batches for your NEET preparation. Clear, bache? Right? He's mentoring the NEET aspirants from many years, bache. Right? He's a rank producer, so he knows what to provide you, what not to. Right? Right? So be the part of this. So you know that what you have to do. In the description box, there is a link given. Okay. So just click on that link and be the part of this particular batch. And do subscribe our channel, because here in this particular, yes, do subscribe our channel, because bache, we are going to provide you everything related to your NEET examination there on this channel, right? If there is any update, you will get to know about it. Okay, if there is any motivation, strategy, academic content, everything you are going to get on our channel. So please subscribe our channel as soon as possible, and share the link with others as well. Okay, so that is what we need from your side. Done? So do it as soon as possible. And what you have to do after the class, in the comment section, just write down your homework answers, and that's it, that's all. And do let me know how was the session. Okay, so I'll meet you tomorrow at 11 AM and we will finish this particular chapter, and then we will practice the questions as well. And the next unit, which unit you want to start next, human health and disease or the genetics, do let me know. So thank you so much, take care, bye bye.